My name is Catherine Liu, and I am a professor at Fred Hutchinson Cancer Center and in the Division of Allergy and Infectious Diseases at University of Washington. I am excited to tell you today about some great opportunities for training in immunocompromised host infectious diseases. Not only do we have a dedicated infectious diseases clinical service for both our cancer center and solid organ transplant infectious disease, but we also have a NIH-supported immunocompromised host ID T32 training grant. The mission of our training program is to develop the next generation of physicians and postdoctoral scientists who have expertise in infections in the immunocompromised hosts. We provide rigorous and interdisciplinary training to foster innovative and collaborative research in infectious diseases in the transplant setting and in other areas of immunosuppression. Our goal is that individuals who leave our program will use their expertise to advance knowledge and develop innovative strategies to prevent and treat infections in this vulnerable population. We have a tremendous group of infectious disease faculty who attend on the inpatient clinical service, but who also have a diverse array of research interests, which we'll talk about, and who are very much dedicated to mentoring trainees in this space. Dr. Michael Book is our program head and an international expert in CMV infections and respiratory viral infections in hematopoietic cell transplant recipients. In addition to our infectious disease faculty, whom you may meet on the wards, we also have a number of laboratory-based faculty who are focused on viral infections, including herpes simplex and HIV, as well as the human microbiota. We also have several hematology oncology faculty as well who have served as mentors for our fellows. In addition to our infectious disease faculty, we have a fantastic group of infectious disease advanced practice providers who are outstanding clinicians and serve as sources of continuity for our complex patients in both our outpatient clinic as well as inpatient service. They also help to manage our OPAT service. We also have two terrific infectious disease pharmacists who I am fortunate to work with not only in a clinical capacity when I am on service, but also as part of our antimicrobial stewardship program. Regarding your clinical training, during the first year, you will have the opportunity to rotate through both the Fred Hutch as well as the solid organ transplant inpatient infectious disease consult service. During the second year, you will have an HIV longitudinal clinic, as well as 3 weeks of inpatient service time. And for those of you who are interested in immunocompromised host ID, you may specifically request inpatient blocks focused on the FHCC or SOT ID service. During your third year, you will have the opportunity to rotate in our Fred Hutch outpatient ID clinic, as well as the solid organ transplant ID clinic at University of Washington. And then we also have a really great opportunity for fellows to choose to stay for an additional fourth year, which is supported through the T32, where they can serve as a junior attending on the inpatient service with a senior co-attending. This really provides a nice opportunity for senior fellows to transition into that attending role. Additionally, we have bi-weekly case-based meetings, our clinical transition meetings, where the outgoing inpatient ID attending presents cases over the past 2 weeks of their service time, highlighting key teaching points as well as complex clinical management cases for discussion with the rest of the faculty. We also have quarterly clinical care conferences, which are focused on ID management issues within the Fred Hutch service. And then finally, for those fellows who have an interest in infection prevention and antimicrobial stewardship, we have a dedicated IP and antimicrobial stewardship program at Fred Hutch, and the fellows have the opportunity to participate in those meetings, as well as in guideline development, as well as implementation of various quality improvement projects. Regarding the research training, there are 3 tracks within our T32. The first track is a clinical research track that includes also opportunities in infection prevention and antimicrobial stewardship. The track leads include Michael Book, who is the program head here at Fred Hutch, as well as Ajit Lemay, who is the director of the Solid Organ Transplant ID Service. And Jeff Hill leads the immunology and immunogenetics track, and David Fredericks leads the microbiome and pathogenesis track. Our research training goals are threefold. First, to develop critical skills in research methodologies that are relevant to each individual track. We also work with fellows to develop skills in presenting research at local, national, and international meetings. And finally, we aim to develop skills in scientific writing, including scientific papers, research proposals, and grants. There are a number of opportunities for fellows to gain additional training skills in research. There is a master's program in epidemiology through the University of Washington. For fellows who have a specific interest in quality improvement, there is a certificate program in patient safety and quality that is available also through University of Washington. And we are very fortunate at Fred Hutch to have a dedicated infectious disease biostatistician, Elizabeth Krantz, who has really been an outstanding mentor for trainees, providing a biostatistical support for their research. We also host manuscript writing boot camps, as well as research and progress meetings, which are informal opportunities for fellows to present their work in progress and to get feedback from other fellows and faculty. And then finally, we have several affinity groups, one in infectious diseases and immunocompromised hosts, and another in mathematical modeling, where external faculty as well as internal faculty from within the system are invited to present their research. And this provides a really nice opportunity for networking for the fellows, as well as an opportunity for them to learn about some of the new research and hot topics in the field. Here you can see a list of our faculty and a variety of research interests, including clinical, translational, and laboratory-based research on viral infections that affect immunocompromised hosts, including CMV, HHV6, respiratory viral infections, as well as novel diagnostics and therapeutic approaches for viral infections and fungal infections, and how the changes in the microbiome may impact the risk of infection and outcomes in stem cell transplant in patients receiving other cancer therapies. Additionally, we have faculty with interests in the area of infection prevention, antimicrobial stewardship, and antimicrobial resistance. We also have several faculty members who conduct their research at the Uganda Cancer Institute, and there are potential opportunities for fellows interested in the intersection between infections in immunocompromised hosts and global health to participate in projects. I encourage you to take a look at our website as well as T32 training brochure for more information about our faculty interests. And to highlight some of the work done by recent fellows, as you can see, we have fellows with several publications on viral infections, including CMV in both stem cell and solid organ transplant recipients, HHV6, and BK virus-associated hemorrhagic cystitis in stem cell transplant recipients. In the area of respiratory viral infections, fellows have participated in projects, including this one by Jim Boone-Yaratana-Cornkit, who is one of our assistant professors now, was a former fellow in our training program who published this paper looking at the predictive value of respiratory viral detection in the upper respiratory tract for lower respiratory tract infection among hematopoietic stem cell transplant recipients. We have also had several fellows engaged in research related to SARS-CoV-2. Here's one on the humoral and cellular responses to SARS-CoV-2 vaccine before and after CAR T-cell therapy. And this was a large multi-center cohort study led by one of our former fellows on COVID-19 in solid organ transplant recipients. Fellows have also been engaged in research on novel infectious disease diagnostics, including microbial cell-free DNA and metagenomic next-generation sequencing-based diagnostics. Finally, there are opportunities for fellows to participate in research related to the gut microbiome. Here is a really interesting paper by a former trainee who found that stool microbiota at neutrophil recovery is predictive of severe graft versus host disease. We've also had fellows engaged in a variety of projects related to antimicrobial stewardship in cancer and transplant populations, including at our Uganda Cancer Institute. The Fred Hutch hosts a biennial symposium on infectious diseases in the immunocompromised host, which draws participants from all over the country and world. These are photos from a very successful meeting we had this past spring, where we had a fantastic program, including faculty presenting on hot topics in transplant and oncology ID, and trainees who presented their research in a terrific poster session. With that, I will stop here. Please do not hesitate to reach out to any of us with questions. And again, for more information about our program, please use the QR code, which links to our program website and brochure.